Hi, everybody. It's Congressman Jamie Raskin, sending all best greetings to all of my friends out in Maryland's beautiful 8th Congressional District, our friends across the state, and our friends across the country. It's Friday, September 26th. It's time for my favorite time of the week, Local Hero, when we get to salute and interact with the people who make life so extraordinary here in Maryland's 8th District. And today we are honoring for his remarkable career as a scientist and in public service, Dr. Eric Green. He's a genomics researcher, a participant in the Human Genome Project, former director of the National Human Genome Research Institute at NIH, where he has spent the last 31 years of his career. He was the third director of the Genome Research Institute, having been appointed by Dr. Francis Collins in 2009. Dr. Green served as director from 2009 to 2025, up until this year, and we'll learn something about his unceremonious departure. But he was at the Institute for over 30 years, during which time he was appointed to multiple key positions prior to becoming the director. For just over 2 decades, Dr. Green directed an independent research program that included integral start-to-finish roles in the Human Genome Project, groundbreaking work on mapping, sequencing, and comparing mammalian genomes, and key discoveries about the genes that are involved in several rare genetic diseases. He got his MD and his PhD degrees in 1987 from Washington University in St. Louis, where he grew up. And throughout his career, he's authored and co-authored around 400 scientific publications and has garnered every conceivable national award for scientists. Following his greater than 30 years of public service as a federal employee at the Institute and NIH, Dr. Green is now using his experience to lead government-funded research programs to foster the expansion and the utility of genomics in academia, healthcare, and everyday life. So he has had this dazzling career in science, in pursuit of humanity in the public interest. Dr. Green, for all these reasons, and for the fact that you've been a resident of Bethesda for more than 3 decades and are a proud Marylander, we commend you and we recognize you as our local hero.
Well, thank you very much, Congressman. I'm delighted to be talking to you. That was a very kind and generous introduction. And I want to thank you for all you do, not only in supporting NIH and supporting biomedical research, but being a great advocate for democracy and representing us so well in the U.S. Congress. I am a big fan, as is the rest of my family.
Well, thank you. That means a lot to me. So look, we're living at a time of these really astounding breakthroughs in science, and particularly the use of genomics in medicine. And you've been such a pioneer in that. And yet at the same time, we're living in a period of profound assault against science. And I want to take our moments with you to talk about both sides of it. Would you start by giving us a sense of the great scientific achievements you've been part of over the last few decades and what's going on now? And then we can talk about what's happening politically.
So I feel very fortunate. I trained as a physician scientist, and I literally graduated the same year this word genomics was coined. The word didn't exist until 1987. And genomics is the study of all the DNA of an organism. So the human blueprint is the human genome. And the field of genomics was named in 1987, in part because we recognized as a biomedical research community that we had the tools in hand to do something unprecedented, to read out the human blueprint for the very first time. And by read out, I mean our DNA consists of 4 different building blocks. We abbreviate them A, G, T, and C. And a human genome can be represented as 3 billion of these letters. And in that is the code for making and operating a human being. And by the late 1980s, when it became clear that, wow, we could read out the 3 billion building blocks of the human blueprint, we got very excited about the idea of launching a big international project called the Human Genome Project. U.S. Congress got behind it and created one of the components of the National Institutes of Health to lead the U.S.'s effort in it. And as a recently graduated physician scientist, I was training in pathology. I had the opportunity to do some research, and I jumped into a laboratory at Washington University that was led by one of the founding leaders of this new field of genomics. And before you know it, in a blink of an eye, I was a trainee working on the front line of the Human Genome Project. And I was involved in it from beginning to end, from 1990 to 2003. In 2003, we finished it. We had read out the human genome blueprint, the sequence of the human genome for the very first time. Now, in the middle of the project, we went from the first director of the Genome Institute, which was Jim Watson, to the second director, Francis Collins. And when Francis Collins was recruited to lead the Genome Institute in 1993, he wanted to create a government-based, within the Institute, research program right there on the Bethesda campus that was going to focus on genomics. And he called me and said, "You want to come join me?" And I was just a second-year faculty member, but I said, "I'm there, Francis, I'll join you." So in 1994, my wife and I moved to Bethesda, Maryland. We built a house 2 miles north of NIH, and I spent the next 31 years there. Shortly after arriving, within a couple of years, I was tenured, so I was honored to be sort of made a full senior investigator in the intramural program. And then I started accumulating leadership positions as opportunities came up. At one point, I actually led the on-campus genomics program in our institute at the NIH. But then when Francis Collins was appointed to be President Obama's NIH director, it left a vacancy at the Genome Institute in terms of the directorship. I applied for it, and I was honored to be selected to be the director in 2009. And so from 2009 until March of this year, I was the Genome Institute's director, and it was a remarkable honor. I feel like we got a tremendous amount done. And where we are now, and it really represents the 15 years, the timing was perfect, is the reason we did the Genome Project in the first place, the reason we wanted to be able to read out the human genome, is that every one of us is incredibly the same in our spelling of our DNA, but we also have quite a few differences. I mean, Jamie, you and I are 99.6% identical across all those billions of letters of our DNA, but that leaves 0.4%. That's different. That's millions of spelling differences between your sequence of your genome and mine. Many of those spelling differences are not consequential for our health and our well-being, but some of them are. And in fact, we are now to the point over the last 15 years of beginning to understand which of those differences are relevant to our health and how to use information about individual patients' differences in the spelling of their DNA to better manage their medical care. That's something known as genomic medicine. And what we've really accomplished at the Genome Institute over the last 15 years is to make that a reality. We went from having a vision that one day we would be able to do this, but now we're starting to do this. It's just the beginning, but it's incredibly exciting and it's game-changing for people. And it's really a time where there is so much potential to have our foot fully on that accelerator, to really see to realize the potential of genomics in improving how we practice medicine, better managing disease, better preventing disease, and better developing new treatments based on that information. And so it was a thrilling ride. I think it's been slowed down a bit now, and I'm very concerned about what the future is going to look like, at least in terms of NIH's leadership in this key area. So that's my story.
All right, so before we turn to these unfortunate sequence of events beginning in January, your career so far then traces this remarkable arc because you were involved in helping, some of your mentors and leaders actually map out DNA, right?
Exactly. Yep.
Figure out what it all was. You guys actually completed the project. The human DNA was totally mapped out. And now you're involved in this process of applying and deploying that knowledge to the treatment of illness and disease and figuring out how to improve medical technology and the medical response to particular problems, right?
That's exactly it. That's exactly where we are. And in so many different areas, whether it's cancer or whether it's for monitoring the health of unborn children, whether it is dealing with children or adults with rare genetic diseases, or in some cases, it's about tailoring which is the best medication to take because so many medications are out there, and sometimes it's just sort of hit or miss. We are learning that encoded in our DNA are spelling differences that dictate which might be the best hypertension medication to take and for you, or maybe what's the best dose. And that's something, big word called pharmacogenomics, but it really is about just improving and optimizing how we practice medicine. So much about medicine, for up until now, we've been flying blind because a patient sitting in front of you, you didn't have an ability to look inside that patient's blueprint, but now we can do that.
All right, so and just say one further word, if you would. This is so fascinating about the role that artificial intelligence might play in enhancing this field of
Genomics. Yes.
genomics. Yeah.
Well, I mean, people always ask about how artificial intelligence is going to be used in genomics. Just imagine that you have billions and billions of letters in your DNA, and then you have some of them that are different between people. And then you can integrate that data with other data, like all the information in your electronic health records, or maybe other information about you or your social environment or your physical environment, so forth. That is a data crunch problem. And just even interpreting a generic human genome sequence, let alone an individual patient's, it is data crunch. And the human brain is pretty good at that, but not nearly as good as if we do it in combination with artificial intelligence. And so there is no question that the marriage of genomics and AI is a marriage made in heaven.
Yeah. So we're living in a time of extraordinary possibility. Okay. And yet, back in the grounded world of politics and sociology and government, NIH has come under literally unprecedented attack in its entire history over the last several months. And I wonder if you would say something in general about the attack on science, the attack on NIH, and then what that has meant for your particular program and your leadership.
Yeah, I mean, it is a full attack at every level. Some of it is specific to specific areas of science, obviously vaccines, things like that, COVID, blah, blah, blah. And then some of it is, and then some of it is about, the idea that government is as waste, fraud, and abuse. And then some of it is just wanting to just reduce the size of the federal workforce and just get rid of these people that have been in the government and try to put in new people. I've seen all these things happen at the Genome Institute, and I was part of it. So my circumstance is that the NIH consists of 27 major components. These are institutes and centers, and there's 27 of them, and each of them have a director. And that is the most incredible group in the world, led by the NIH director. And I was so fortunate to be part of that group at 27 for 15 years. This is the brain trust, in many ways, of biomedical research for the whole world, since the NIH is the crown jewel of biomedical research, the envy of the world. And we are appointed every 5 years, and then we get reappointed if we're doing a good job. I was up for my reappointment. I was reviewed back in December. I got a very positive review. But I drew the short straw of having the first reappointment date for my next 5 years, the very first one after Inauguration Day. And so it came time for me to be reappointed in the middle of March. And
you were ready and willing and eager to keep going.
Oh my gosh, yes. I mean, we had tons of stuff to do. We had to, absolutely. And I was just told they weren't going to reappoint me. And it was a little bit of back and forth, but ultimately they said, we're not going to reappoint you. I was retirement eligible, so I formally retired, but it was a forced retirement. It was not by choice. I didn't even know how to spell retirement. It's not in my DNA to retire. I'd like to work. I love this incredible.
Your DNA is not spelled like that.
No, it's just not spelled that way. So I was forced to retire, at least from federal service, certainly not from being in the field of genomics. So that happened to me in March, but we had already lost some of our probationary employees.
Do you think there was a specific reason that they wanted to force you out substantively, or do you think you just happened to be the first sacrificial lamb?
So as far as I know, and we don't totally know all the details, from everything I've learned, I think I was an easy decapitation of one of the 27 leaders because it was convenient. It was an inaction, and I just had it was bad luck. And the reason I think it is because then they subsequently, a couple weeks later, maybe a week later, they did terminate my deputy director. And then they did terminate about 5 or 6 of the other institute directors, but all of those have hot button things associated with them in terms of what are controversial in the new administration. So, I was the first and I was easy. So I don't necessarily think it was personal, but I guess I won't ever know. But then on April 1st, I watched, it was horrible because that was the big day of the reduction in force. And I saw major groups that I had built up and was so proud of and were just the absolute best at the NIH, like our entire communications group, our entire education group, our entire policy group, our entire workforce development group, they all got terminated. And that was a very dark day at the NIH. And it was dark even for me, even though I wasn't working there, because colleagues that I had recruited, programs that I had nurtured, seeing the best in class work that they are doing, and they're just now all gone. And it's really quite sad. I'll give you one of many examples I can give you of how sad this is that we had an incredibly valuable website that we had been nurturing and really enhancing that helps communicate the science of genomics to the world. It's genome.gov is our URL. It's very easy to remember. And that website has not been updated since April 1st. Because there's no communication staff to update it. And it has educational materials and patient information and all sorts of things to help communicate this incredible science. And that's just one of many examples.
Yeah, and the apparatus of NIH includes not just the world's leading pioneer scientists, but also people who are involved in communicating it, translating it, of all kinds of other work.
Educators educate about it. It just goes on and on and on. So that was one thing. And of course, there's been an assault on DEI work and diversity work and disparities work. And so I also saw a lot of our scientific programs canceled or compromised or halted. And so that's and then there's the other thing that's happened with all these incentivize and reduce the force. A lot of the apparatus to get money out to the researchers across the country to do this work has been compromised because we don't have as many staff to do that because some of them they were either probationary, they got let go, or they were incentivized to retire. On inauguration day versus now, there's about maybe 120, 150 fewer people at our institute, and it's almost like maybe 20%. I don't know the exact number, but it is just decimated our workforce.
Well, this is a story not just at NIH, but also at USAID and the Consumer Financial Protection Bureau and the State Department, the Education Department. Maryland has lost more than 15,000 federal workers who've been fired, riffed, demoted, forced out of their jobs, and so on. So it's just an absolute gutting of the federal workforce, which is so critical to all of these public projects. So, look,
Congressman, I want to point something out. So we have a lot of replenishment we eventually need to do, but replenishment happens when you have strong leadership. Knowing I was going to talk to you today, I looked it up, and out of the 27 institute directors that sit around this glorious table and really are the brain trust, out of 27 of them, 14 are acting. In other words, there was some already vacant, but now I started a whole, I was the first of a bunch that got released, and now some others have just left and retired and so forth. And I know of at least one other who's likely going to be leaving. So these people you need to replace the leadership. This will not happen overnight. This is going to take years to be able to do this. And I very much worry about sort of the rebuilding when we are so short of the highest level leaders to help do it. And it's not just the very top leaders, it's the next levels down, the best of the administrative staff, the best of the scientific leaders that run the on-campus program. We have a lot of vacancies, and I'm a little worried how easy it's going to be to get the best of the best to come into the government, especially at this time.
Yeah. Well, if we've gotten rid of a lot of the very finest scientists and administrators like you, and then you put in people who may be political scientists and not scientists, or people who are there for another reason, and then you're trying to recruit from people who are the brilliant young people, but now suddenly they're skeptical of going into the NIH and going into government work because of what's happened. It really is like throwing on the brakes for scientific progress at a time when we could be doing so much to help so many people.
And that's what's so tragic.
We are nonetheless, of course, overwhelmingly grateful to you for everything you've done as a scientist and also for speaking out about what's taking place and how harmful it is to the public health, to science, to the integrity of government right now. And so, for all these reasons, we want to commend you and salute you and make you our local hero. And you get to have the last word.
My last word is I remain optimistic because I see this incredible ecosystem of biomedical research that has been the envy of the world here in the United States. I think we've hit a pretty significant series of speed bumps. I'm going to keep my fingers crossed that people like you and others in the Congress and others around the country are going to recognize how, I know you believe it, but others will recognize how important it is to have a healthy and robust NIH. I think we need to take a step back and realize we need to rebuild and replace and get to the point of having the NIH that we had, prior to these destructive forces. And, I really want to see us rebuild. We have some incredible things that can be accomplished if we have a healthy NIH. And I want to just emphasize, I will help in any way I can at any time when the NIH rebuilding begins. I actually, still have my house in Bethesda, and I want to help give any kind of advice I can because I want to see NIH thrive the way it once did.
That's our local hero, Dr. Eric Green, a 31-year veteran of NIH and all kinds of amazing breakthroughs in the field of genomics and now working in the field of medical genomics, and also standing up for science and for everybody at NIH who's working on it. Thank you, Dr. Green, and please accept our warm thanks, and I hope everybody will bombard you with their love and congratulations.